Hello, and thank you for joining us for our mini lecture about protein synthesis. Proteins are a vital part of our bodies. They make up most structural components of our cells and aid in nearly all of our cellular functions. We previously learned about DNA in our DNA and mitosis video. Our DNA contains the instructions for how to produce every protein in our bodies. If you recall, our DNA is made up of genes. Each gene contains the genetic code to build a single protein. Gene expression converts the information stored in a gene into its final product, ultimately shaping the cell's structure and function by controlling which proteins are produced. Proteins are built from amino acids. The genetic code is made up of triplets, which are sections of DNA made up of 3 bases. Recall that the bases are adenine, thymine, cytosine, and guanine, A, T, C, and G. The triplet sequences each code for a single amino acid. A gene, which is composed of multiple triplets in a unique sequence, provides the code to build an entire protein, which is made up of a chain of amino acids in a specific sequence. You may recall this image from the DNA replication video. Notice down here at the bottom how the nucleotide bases form the 2 strands of DNA. This is the sequence that will code for the production of proteins, with 3 bases, like TAC, coding for a single amino acid, and multiple triplets coding for a full protein. The synthesis of proteins occurs in 2 phases, which we will look at next. The first phase of protein synthesis is transcription, where the DNA code is transcribed into RNA, specifically messenger RNA or mRNA. This is very important because the DNA is contained within the nucleus of the cell, but protein synthesis occurs in the cytoplasm of the cell. Messenger RNA brings the DNA code from a single gene from the nucleus out to the cytoplasm. Note that messenger RNA is a single strand, unlike DNA, which exists as a double strand. One important thing to note about RNA is that it does not contain thymine. Thymine is too large to fit through the nuclear pores, which is how the mRNA exits the nucleus to get to the cytoplasm. Instead of thymine, RNA contains uracil as a substitute. So where you see thymine here is going to bring in adenine, the complementary base. Adenine on the DNA strand is going to bring in uracil in the messenger RNA strand. Transcription looks a bit like DNA replication, where a region of the DNA will unwind and the 2 strands will separate. Transcription then takes place in 3 stages. Stage 1 is called initiation. Each gene in the DNA sequence contains a promoter, which is a particular sequence of nucleotides that triggers the start of or initiation of transcription. Stage 2 is elongation. The DNA strand is unwound by the enzyme RNA polymerase, which you can see here. The coding strand of the DNA becomes the template with the genes that are to be coded. As the RNA polymerase moves down the template strand, it assembles the messenger RNA with the complementary bases to that template strand, substituting uracil for thymine. So for an A in the template strand, a U will be added to the messenger RNA strand, like we saw here. For every C, a G is added, and vice versa. So GC, CG. After the RNA polymerase has moved across the entire gene, the final messenger RNA strand is created. Stage 3 is termination. At the end of the gene that is being coded, there is another set of bases called the terminator sequence. Whereas the promoter sequence started transcription, the terminator sequence ends the process by causing the new RNA to fold up on itself, therefore separating it from the DNA strand. This new strand is now called pre-mRNA. This is because it has long regions that do not contain codes for amino acids. These regions are called introns. Those sections need to be removed through a process called splicing before the final mRNA is ready to leave the nucleus. Splicing is the process by which the unnecessary parts of the new pre-mRNA strand are removed to create an mRNA strand that only contains the instructions to code for the protein. A spliceosome attaches to the pre-mRNA strand and cuts out the non-coding segments, or the introns, which you can see here. What's left are the exons, which are the coding segments. There is some variability in this process, so sometimes coding strands are also cut out along with the non-coding strands, and this accounts for some of the variation in proteins that are produced. Once the final messenger RNA strand is complete, it leaves the nucleus through the nuclear pore and enters the cytoplasm of the cell. In the cytoplasm of the cell, the second phase of protein synthesis occurs, which is translation. The messenger RNA strand is made up of codons. A codon is a 3-base sequence of messenger RNA that codes for a specific amino acid, which you can see here where it says codon. AUG is 3 bases that are going to code for a specific amino acid. This chain of codons must now be translated or read in order to assemble the chain of amino acids, which is also called a polypeptide, that will eventually become our protein. There are 2 important molecules that are needed for translation. These are 2 other types of RNA. Ribosomal RNA, or rRNA, composes the structures of the ribosome. Remember that ribosomes aid in protein synthesis. Ribosomes consist of both a small and large subunit. When a messenger RNA molecule is ready to be translated, the 2 subunits come together and attach to the messenger RNA. So you can see that at the top here, where we have the small ribosomal subunit and the large ribosomal subunit coming together so that they can read this mRNA strand. The other type of RNA that is essential to this process is called transfer RNA, or tRNA. And you can see that here. tRNA brings the amino acids that each codon on the mRNA strand codes for and attaches the new amino acids to the building polypeptide chain. The tRNA has a 3-base sequence at one end that matches the codon on the mRNA strand. So here we have AUG, and that is going to match with this complementary sequence UAC. So it's on the mRNA strand, it's the codon, and on the tRNA strand, it's called the anticodon. On the other end of the tRNA, it contains a binding site for the amino acid for the codon that is complementary to the anticodon on the tRNA strand. So here's the binding site on the tRNA. So you can see in this middle, the codon AUG on the messenger RNA strand attracts the anticodon UAC on the tRNA molecule. This tRNA molecule then holds the amino acid methionine. Translation also consists of 3 main stages: initiation, elongation, and termination. Initiation occurs with the binding of the ribosome to the mRNA, which we saw up here at the top. Elongation occurs as the ribosome reads the messenger RNA sequence and the tRNA brings in the correct amino acids, thus forming the polypeptide chain. Note that this stage does require energy to occur. As each tRNA brings and attaches its amino acid to the polypeptide chain, the ribosome then moves down 1 codon, releases the first tRNA molecule, and calls in the next one. So here, this first tRNA is getting released, this one is still attached, and the third one is coming in as this ribosome moves down the messenger RNA strand. Here, the first tRNA has left methionine, and now the second one is attaching the phenylalanine, and the third one is bringing the third amino acid. Termination occurs when the final codon of the messenger RNA strand is reached. This is called the stop codon, and it signals that translation is complete and triggers the release of the newly synthesized protein. Let's do a quick review. Protein synthesis is a multi-step process. Our DNA contains the instructions to create every protein our body needs. The DNA has to be transcribed into messenger RNA inside the nucleus of the cell. So that's happening here in the nucleus. During this stage, thymine is substituted with uracil, so the messenger RNA can fit through the nuclear pores. The messenger RNA then leaves the nucleus and moves into the cytoplasm, here, where translation occurs. During translation, the ribosome reads the code on the messenger RNA and uses transfer RNA to assemble the polypeptide chain that will become the final protein. Thanks so much for watching our presentation on protein synthesis. We'll see you back next time.